All right. So this is chapter 3, and we are starting off with talking about the membrane junction. As you know, there are ways in which the cells are connected with one another. The cells connecting with one another for structural support purposes, for maintaining, and for further stability. So tight junction uh, is kind of like a intercellular protein that we discussed. These are between the two cells, and it helps to connect the cells together, but nothing really pass. So let's go take a look. Right here, it looks like it's one cell. Here is another cell. So if you wanted to kind of like draw it, and right in between them, right here, this protein, which again is being expanded right here, these are the junctional protein. These are called tight junction. It's called tight junction because substances cannot go through these. If you have, like, let's say ions, sodium, potassium, things like that, they cannot pass through. All right, these are closed. All right, but they are adhering to one another, so that way the cells are kind of like connected to one another. All right. Next up, you have desmosomes. Desmosomes, there are crisscrossing linker proteins that are also attached to the intermediate filaments indirectly. And the intermediate filaments, they're going deep and holding on to these organelles and uh, anchoring them. So this is called the desmosomes. Now, why do we need to have a desmosomes, and which type of cells do have desmosomes? Cells that are subject to high mechanical degree or stress, they are usually have a lot of desmosomes. This could be the palm of your hand, for example, or bottom of your feet when you are jumping up and down, walking, things like that. You need more structural stability. Think of it as perhaps maybe you know you're anchoring something like a cabinet or whatnot, but you're using very, very long screws, and you're screwing the cabinet into the wall very deeply, and the screws are kind of like these filaments. All right, and these cells are kind of like cabinet. It allows it to have more stability and structural support due to the nature of these cells, because they're subject to more mechanical stress. The third and the final membrane junction, you have a gap junction. The gap junction, there are tiny little spaces for it. So in other words, these are kind of like, if you wanted to think of it as in terms of function, because they are on the membrane and so on, these they have a function of transport. Earlier, we discussed about 6 different function of the protein. Let's go and review that really quickly. Uh, if you recall, we mentioned that some of these proteins on the membrane, here's the membrane, they are acting as a transport. In other words, we are bringing in or out substances through these proteins. Other ones, they had enzymatic activity. Other ones, they were kind of like a receptor. Other ones, they were kind of like intercellular joining. So this was kind of like similar to the tight junction. Cell-to-cell recognition in immune system, and again, attachment to the cytoskeletal. This is kind of like a desmosomes. But what we're looking at mostly right now is kind of like this. So these are called gap junction. The gap junction, there are tiny spaces where substances could actually pass through these small pores in them. So what does that do? Let's say this is the intestinal walls, all right? So you absorbing some material, say a particular amino acid is going in, all right? And you had enough, or water, let's say. Now they can go through this gap junction in the nearby cells, and the nearby cells can have extra water, or whatever the case might be, all right? So there's a gap in there. Interestingly enough, Our heart cells have these gap junction as well. The electrical impulses can go through this gap junction, and as a result of that, the whole heart can actually contract or relax. All right, next up, we're going to go ahead and take a look at the system of transport. Now, in general, we have two system of transport, okay? One happen to be passive transport. Passive transport, no energy is required, which include diffusion. And there are different kinds of diffusion. There is a simple diffusion, there is a facilitative diffusion, there is a carrier mediated diffusion, or channel receptor mediated. So there are different category of it. Simple diffusion, you have just a regular diffusion, simple diffusion, you have facilitative diffusion. All right. And we got osmosis. Osmosis movement of the water is also considered to be uh, that as well. Okay. And also filtration. Filtration, I'll talk about it. It really depends how you look at it. And then you have an active transport. Active transport requires energy. So this doesn't need energy. Active transport requires energy. All right. And again, there are different types of active transport, and we will take a look at different type of transport. Primary active transport, secondary active transport. We will discuss perhaps vesicular transport, which includes the endocytosis and exocytosis. All right. So what I'm doing is just giving you guys the outline of what I'm covering right now, and then we can go from there. Then you have receptor mediated. There are different type of endocytosis. We will take a look at probably. Let me erase this thing. There are different kind of endocytosis. You have uh, pinocytosis. You have phagocytosis, right? Uh, Pinocytosis is also known as a fluid phase, and you have phagocytosis. And finally, there is a receptor mediated endocytosis. So majority of these obviously are requiring energy, and we will discuss about these and go over the details of those as well. All right. So interestingly enough, they required ATP. All right. So right now we are going to the passive process, no energy needed. All right, let's go ahead and take a look at the diffusion. What is diffusion to begin with? Diffusion is basically substances from high concentration, bracket means high concentration, arrow means high, goes to the area with a lower concentration. This is called diffusion. All right. So let's say that you have a beaker full of water, all right? And this beaker of full of water, you put a drop of, let's say, red ink inside. All right. As soon as you put in the drop of ink, if you kind of like swirl it a little bit, you notice like, the higher concentration of the red dye or red ink in this case is going to go to the area with the lower. So very soon, after some time passes by, your beaker is going to be all red, right? All right. Why? Because again, they went from high towards the area that is low. So that was a diffusion. Now, the next thing that I wanted to talk about is what is simple diffusion? Simple diffusion is the same concept, folks, except the fact that you need to have the membrane around it. So, for example, right over here, we can go ahead and put in like some kind of a membrane in here. It still is favorable. Things are going, substances are going from high concentration towards the low concentration, but membrane is required. All right. Say, for example, you have a balloon that has a helium in it. All right. And after some time passes, the balloon is going to be down on the floor. What happened? Well, helium that had higher concentration in here went to the area with the lower concentration. So they seep outside because there is lower concentration of helium. Over here, it was a high concentration, but they had to pass through this wall. In this case, the membrane of the balloon wall is a sort of a membrane, right? So this actually very important because when we're looking at our cell, all right, uh, you'll have higher amount of oxygen in the blood vessels, let's say capillaries, all right, and you'll have lower concentration of oxygen inside of our cell. So oxygen goes from high to low. They're passing the membrane, all right. Simultaneously, what happens is inside of the cell, carbon dioxide is high, and in the blood, carbon dioxide is low. So again, they're going and exchanging of the gases can occur this way. All right. So in the cells of the lungs, it would be opposite.
Professor, we can hear you.
Oh, I'm sorry. I accidentally clicked on the mute button. I appreciate that. All right. So we were saying that this is the simple diffusion, you guys, and the simple diffusion requires the substances to pass through the membrane. uh, Oxygen and carbon dioxide going back and forth, passing our membrane is a good example of the simple diffusion. Now, I was saying and explaining that, don't limit your knowledge to just the examples I give you. Google it, different examples of simple diffusion, write it down. And there you open up another door to serve yourself to understand the material even better. All right. So sometimes you may see a different scenario that the concept is what we talked about. However, the examples might be different than what you hear on the lecture. So I don't want it to give you guys a panic mode or anything like that. It's just I wanted you guys to emphasize on learning, not memorizing. Anatomy is done. That was a lot of memorization. All right. But in physiology, you really need to get this stuff down. All right. So now we're going to go over the facilitative diffusion. What is facilitative diffusion? Let's go ahead and erase some of these parts so we could get that out of the way. Facilitative diffusion requires a transport protein and the membrane. So requires transport protein and the membrane. So for example, right over here, I would say that you have some type of a protein, and this like a membrane, right? These are the protein, and substances are going right through this protein. These are carrier protein, facilitative diffusion. Now you're still going from high to low, all right? So let's say, for example, you have a cell, all right? And the cell has a transport protein in here, all right? And outside of the cell, you have higher amount of sodium, and inside of the cell, you have lower amount of sodium. Sodium has a polarity, it has a charge, and this charge prevent the sodium to be able to go inside. Sodium is not permeable to go inside. Yes, they want to go inside, they're high. In here, they're low, but unfortunately, they cannot. All right, why not? Because again, they're charged substances. Charged substances, they have a hard time passing through because remember, the layer in the middle of the membrane was lipids. They don't pass through the lipids. Lipids are non-polar, all right? So they can't. However, if these channels are available, and there's more than one, I'm just showing you one, then they could go through these and go from high to low, all right? So what we say is these carrier proteins or transport protein, they facilitate this. So this is known as facilitated, facilitated diffusion. Doesn't need any energy in order to do the facilitative diffusion. All right, we are done with all kinds of diffusion. Next thing in line, I would like to go ahead and give you an idea about osmosis. Osmosis is related to the water movement. Now we are only looking at the water movement, all right? So let's say that you have a YouTube. Now this is a different than the YouTube videos, all right? And let's say that you have a membrane in here, all right? And you have two sides. Let's say this is side A, this is side B, all right? And let's say that this is freely permeable uh, to freely permeable membrane to solutes and in this case water solvent. And let's say side A we had, let's say, some solute, say glucose. All right. Now you can see what's happening. These glucose, because this membrane is free, they could go to the other side. Well, you might say, well, this is simple diffusion. Yes, because glucose is going from high towards the area that is low. That's the side B. All right. However, what if I change this freely permeable membrane into selectively permeable membrane? Let's say I have a capability or I make another YouTube and now I'm changing these membrane. I'm making it to have it a smaller pores. Now they cannot pass through, you see, because it's not big enough, they're not large enough for them to pass through. They can't. However, in this case, you have more water on this side, on the side B, because there is no more solute. So you have more water on this side. So if you have more water, what happens? You have more water in here, you have less water in here, all right? And the water is permeable because it's selectively permeable. That means some stuff can pass, some stuff is not, all right? So now water can go from side B towards the side A. And this part right over here is probably rises, and this part right over here, it dips, all right? So that's what happened. Movement of water towards uh, solute is called osmosis. Passing the uh, passing the selective permeable membrane must pass. All right. Uh, so passing selective permeable membrane. So from here it comes here. All right. It's called osmosis. And osmosis is all around us as well. A lot of times, the protein, there are uh, tiny little protein in our cells that allows the water to pass. All right. And for example, you have a cell, you might say, wait a minute, water is polar. They cannot pass in here. You're absolutely right. I mean, they're polar. Why would they be able to pass the membrane that is non-polar? You know, uh, but there are tiny little channels in there that we call these channels aquaporin. So when you have a higher water, for example, and you have higher solute on this side, then that's what happens. Water can go towards the area with the higher solute, and in this case, water is going to go in. All right. So homeostasis and keeping things balanced is really important. Now, if you have a lot of solute on one side, let's say we increase the amount of sugar on this side and so on, the osmotic pressure now is going to be higher because it's pulling more water on this side because there's more solute there. All right. So this is rather interesting. And a lot of times, uh, our blood pressure is being maintained this way too, by the way. Let's go take a look. Here you have a blood vessel, all right? And let's say this is a capillary. Now, in anatomy, you learn from arterioles goes into the capillary, and from capillary goes into the venules. From venules goes into the veins. Before arterioles is artery. So let's say the arteries is larger diameter. Blood is coming in. Blood contains water. But when it gets to the capillaries, the diameter is going to get really small. And some of the water is going to exit. All right. And now there are other stuff in our blood like white blood cells, red blood cells, protein molecules, ions of all kinds and so on. So this allows some of the water that leaves to pull back in. Why? Because as water leaves, there's going to be higher solute in here now. So now in the capillaries, what happened is the water kind of like get pulled back in so you don't lose water. All right. Now in here you have proteins, in here you have ions, you have calcium, you have sodium, you have potassium, we discussed this, we have glucose, any imbalance of any of these is going to mess up the osmotic pressure. I'll give you a few reasons. I give you a few examples. Pay attention. This is a real example, folks. All right. Say, for instance, you're taking up way too much sodium chloride, right? Sodium chloride was a salt, if you remember. There was a ionic bonds in between them. All right. So in the solution, they become sodium and chloride ion. They call it dissociation. Basically, I'm going to show that in terms of dots. What is this salt? Remember, the salt is a solute. So when you're taking up a lot of salt, that means your solute inside of blood is going to be higher. Now, the arteries and arterioles, they have three thickness in there: tunica media, tunica, externa, tunica interna. So they're really thick, and water cannot go in or out. But once you get to the capillaries are really thin. If you remember, they were simple squamous epithelium and all that. So they're really thin and permeable to water. So water is leaving, but then this solute is going to be increased, so the water is coming back. However, if the solute already very high and now it's going to get even higher, what will happen? Too much water is going to get going. So here are cells, for examples. All right. So from our cells, water is going to be leaving as well, causing a couple of things. One, you're losing water in your cells. So as the water leaves, you're getting dehydrated, right? This is the reason why it's not a good idea if, let's say, you have no drinking water to go and drink seawater. Seawater or the ocean, water from the ocean, has a high sodium chloride. Now you might say, wait a minute, how does a fish live in there and so on? That's because their system is a little bit different. They can have more concentrated urine and get rid of the urine, and therefore they can balance, and homeostasis is a little bit different than ours. All right. But anyways, a lot of water is going to go in here now. One, you're going to cause low water inside of our cell, causing a dehydration. So the more salt water you drink, the more dehydrated you will get. So it's actually a more worse effect is going to occur. Another thing is now that the water is inside of the blood, our blood pressure is going to go higher. This is the reason why people have a already high blood pressure. They should avoid salt. All right. Because of all this water is coming in, you're increasing the volume. Increasing the volume is going to cause the increasing of pressure. So if you have a faucet and you turn it one time, you're looking at the pressure in here, all right? And you have the same faucet, but you turn it like, let's say, 5 times, and take a look at this water is coming in with a lot of high pressure. All right, so more water is coming in with high pressure. Okay, you could test that in and so on. Well, what did we do? Well, we increased the volume in here. Right, same thing in here. The volume is increasing because you're losing water, but it's going to go into your blood. All right, same thing. All the other ones are similar idea. You might have heard about hypernatremia. That's sodium, excessive amount of sodium. All right, or hyponatremia, lower amount of sodium. So you could see the complexity, but yet very effective way to achieve homeostasis is understanding of the osmosis and what is happening. Now, let's go ahead and take a look at the next phenomena, which is called the tonicity. All right. Uh, tonicity is how the different uh, changes that are occurs, and it can affect the volume of the cell. All right, this is related to the tonicity, ability of solution to change shape or tone of cells, all right, based on the internal water volume. So let me give you an idea, if that didn't make sense. Here you have a red blood cell. So normal volume is normal, everything is good, all right? However, this is from the side view. Red blood cells are concave a little bit. This is from the top view, right? This is from lateral view, side view, right? However, if you take a water from the red blood cell, they might kind of like crenate. They lose their shape. Tonicity is different now, all right? If you put in more water in it, probably they're going to get expanded and so on, and eventually they're going to pop because water, there's a limitation of how much water you could put in there. Our cells are not like balloon with a lot of elastic fibers so that they could expand. It doesn't work that way. All right. So at one point in time, they could pop because again, that's changing the shape and so on. So there are three solutions that can do these three things. One is called isotonic. Isotonic, the amount of solute outside of the cell is similar than inside of the cell. So for example, let's go ahead and take a look at isotonic. There are solute inside of the red blood cell, all right? And therefore, if the solution that you put in this, let's say you put it into a solution or the environment that this has the same as inside, the water going in and out would be the same. So in other words, there is no change in tonicity and there is no change in shape. This is called isotonic solution. So our cells has about 0.9% NaCl, all right? And if you place this inside of the solution with 0.9% NaCl or around there, nothing will happen. In fact, when people getting an IV because they need the blood, the IV is a solution because let's say a person, losing blood, right away they give them an IV to bring back up the pressure, all right? Because as you know, when your blood vessel has a tear in it, so you're losing a lot of blood in here, all right? And for that, you need to inject the IV so that way you monitor and you, of course, you wanted to put pressure in here or possibly block it, depends on how the severity of the damage. But the point is, this IV or a saline solution has the same concentration as inside of the cell, 0.9% NaCl. So in this case, it is not going to affect the cell shape, and that's what we want. However, what if we put the red blood cell inside of the solution which has higher amount of solute? Then what? So let's say you have a beaker and in here is a 10% salt solution, 10 gram per 100 milliliter of water, let's say. All right. Now that's a lot of salt. And if you put in the cells in here, because there's a lot of salt in here, a lot more than what amount of solute inside, then water, where does it go? It always goes towards the area with the higher solute. So water is going to leave. And then as a result of that, a little bit later in time, is going to shrink. So I'm going to show you a little bit later on, is going to shrink. So the tonicity is changed. Such solution are known as hypertonic. Hypertonic are the solution with the higher concentration of solute, and it's going to cause the cell to shrink. All right, last one. Let's go ahead and take a look at hypotonic solution. Hypotonic solution, let's say you have a beaker with pure water. There's no solute at all. And you're placing a red blood cells in it with some solute. Now, because there is amount of water that is high, water is high a lot in here, and then they're supposed to go where? They're supposed to go right through those aquaporin that we discussed, and is going to go right inside, all right, where the solute is. In this case, it's only inside, all right? So what it's going to do is going to pop this cell. So a little bit later on in time, you're not going to see anything. So I'm just going to erase it, all right? So this is called the hypotonic. Our cell does not do very well in the hypotonic, does not do very well under hypertonic, but it does do well under isotonic solution. All right. Let's give you a scenario uh, to see if you understood anything. I could give you the same lectures in probably half of the time if I don't give you examples, if I don't give you, things to solve, if I don't give you, activities to think about things. I could just tell you this is this and then keep going. Why do I take the time? So you could understand it better. However, there is a price that we have to pay now, and that is the shortage in time. All right, because the class is going to be over in a certain amount of time, right? But I would rather do this one, and we might have to kind of like uh, record and place or do something about it later on. But anyways, let's go ahead and take a look at the cell. Here's our cell, all right? Say this is a virus, coronavirus, any virus, lands in our cell, and with these spikes, it lands, all right? Once it lands, it gets in. Now is inside. Now there are different type of virus. I don't want it to teach you virology, RNA-based, DNA-based, retroviruses, all kinds of different things. But let me just in a concise way, viruses use our DNA, our enzyme to make more of what they need. And then these proteins that, I don't know if this is going to be too advanced for you or not, but let's say this is our DNA, double helical structure. And some of the viruses, they can go in and become part of our DNA. So when we are making the RNA or we're making the protein, for example, that's genetic material, right? This section that belongs to the genes for the RNA, for the viruses, regardless if it's RNA or DNA, like I said, there are different type of viruses. You're making proteins now. I already know how you make the proteins. I showed you a little bit of it already. You put in the chain of amino acids together, and then you're making a protein. All right. So messenger RNA, there are read three at a times. You look up that universal coding system, and you come up with the protein. But these proteins belong to the viruses. So very soon, more viruses are going to assemble. Here's what happens after a little while. After a little while, a cell lice, and all these viruses, they get out and they go and infect more cells, regardless of what. That's not important what kind of a virus it is. One time I gave an example of adenoviruses, and the students were panicking. Oh my God, how come he's not covering what we're supposed to? I thought this his class was physiology. Oh my God, please try not to be negative, folks. All right, look at the overall picture. I don't understand. People always have something to nag about. All right? Not everybody, but I never wanted you guys see on the chat, I am confused. How come he's going? Don't ever put those things in, because if you do, then I'm going to take away the chat. And I don't like to hear those things at all, because the way I look at it, you're putting, negative thoughts, and you're preventing and affecting other students as well. You need your full concentration. Right here. What do you think it's? Let me pause the video. If you're listening to this at a later time, think about how this is happening and think about how is it related that to what we discussed. Let's go ahead and take a look at filtration. During filtration, again, it really applicable to the kidneys. You have blood goes into the renal arteries, and renal arteries, there's a kidney and the ureter. So materials that are being filtered, they're going down, and this is called filtration. Now, originally, this filtration, the arrival of the blood in here, that actually requires energy. That's the contraction of the heart. But as soon as they're coming in and just get filtered, then that does not require energy, and that's called filtration. Think about how you filtered your coffee. All right, you put the filter paper and the coffee granules, all right, and then water, and the water just like goes and filtered right through your filter paper, and you collect it on the other side. You really didn't need to have a user energy. So we are done with the passive transport. None of these requires energy. They're all pretty much favorable to go from one to the other. All right. So let's go ahead and take a look at active transport now. Let me just really quickly see what time we have. Okay. Uh, let me pause a little bit, and let's say you got stung by the bee, and the venom is inside now. Okay. Now use a credit card and you take it out the, the sting. A lot of beekeepers recommend to right away, if you have access to honey, put honey in it on top. Some people, they say lemon juice. Here's a lemon juice. You put lemon juice. All right. One at a time. What do you think the honey might do? How is the honey is going to be helpful? What do you think? Let me go ahead and why do you think the honey might be helpful? You guys wanted to, or why do you think the lemon juice, if you juice the lemon on top, might be good? So lemon juice is actually has something to do with acidity. Venom made out of protein. How is the acidity is going to affect the venom structure? All right, make a big deal. So if you apply a lemon juice, for example, the pH is very low, and that's going to create problems in terms of the structure of the protein. So it would affect this venom. It would kind of like alter the shape, and it would be helpful. How about honey? What do you think honey? How is honey is helpful? What do you think? Anybody want to guess?
Maybe like antimicrobial, like a barrier.
No, no, it does have antimicrobial, but this is venom. It has nothing to do with that. Okay, let me go and tell you. Now, honey,
The high sugar content?
yes, high sugar content. What about it? Higher solute, right? Sugar is solute, right?
Yeah.
Solvent is the agent being dissolved. Solute is the agent being dissolved, and solute agent being dissolved, and water was a solvent, is doing the dissolving. So in this case, because it has higher solute, some of the water on your cells, they have a tendency to go towards where? Towards the solute, and hoping that some of this venom is going to come out as well. So it might be helpful. What is this phenomenon? It's osmosis. That's what it is. All right. Let me go over active transport then. We are done with this section. And we are going to part 2, that is part B. And that is called active transport system for now. Okay. So on the active transport, they require ATP. Energy, usable energy is called ATP. And there is something known as sodium potassium pump. So if this is a cell, usually outside of the cell you have higher sodium, inside of the cell you have lower sodium, and you have higher amount of potassium inside of the cell, and you have lower amount of potassium outside of the cell. And there is a special protein in here, and this protein does a lot of different things, but it has different name to it too. But it's called sodium potassium pump. So what it does, it pumps 3 sodium out and brings in 2 potassium. All right. Sodium is going from low concentration to a high concentration. So that's not favorable. And look at the potassium. Potassium going from low concentration to the area with a higher concentration. So again, it's not favorable. But in order to do that, you need ATP. You need to break down the ATP. As the ATP breaks down, that releases energy, and that is allowed for this sodium potassium pump to transport these substances back and forth. All right. This is called primary active transport. Primary active transport. Anytime substances are going towards opposite direction, so in this case you have sodium going inside, going outside, and potassium is going inside. So they are going into a different direction, right? The direction for sodium movement and potassium is different, it's opposite. These are called uh, antiports against one another. Symport is same direction. Movement is same direction. And this one is opposite direction. Okay. Now, what is secondary active transport? Secondary active transport could get a little bit complicated, but pay attention. You might be able to figure this out on the first shot. If not, maybe you'll have to understand this by reading the material a little bit more. All right. So look, I need a space to do this, but let's see if we can. Let's say that you have a cell And the sodium potassium pump is here. So you pump in sodium out, all right, and potassium in. If you pump in the sodium out, what does it do in here? It lowers the sodium, right? Because you're just pumping it out. Right now, on this side of the cell, let's say there is a blood vessel or something that has higher sodium content, all right? And now you have lower sodium in here. There could be a protein channel, transport protein channel, and sometimes these are referred as co-transport, like co-worker, because it can transport two things. Not only sodium can go in, but along with it, amino acid, for example, could go in as well, or sugar can go in as well. So this is called a secondary active transport. Why? Why is it active? You might say, well, I thought you're going from high to low. That should be favorable. Not necessarily. Because the driving force of these going in is this one right here. All right, is this one right here. There are lower amount of sodium now that causes the pulling of this, and along with it, it drags another molecule with it as well. So this is called a secondary active transport. This happens in the kidneys. I will show you a little bit later on the details of it. Okay. Now, let's go ahead and take a look at another scenario. A lot of times, I need more room. A lot of times, there is a phenomenon known as vesicular transport. All right. This is a fluid containing large particles and macromolecules are transported across the membrane. You know, they are inside of the bubble-like structure, sac, known as vesicles. So uh, sometimes, for example, in the nerve, you know, there are proteins or chemicals placed into a bubble-like or a vesicle, for example, right here. This is a good example of a vesicle, all right? And they can be transported down to the axon. Again, if you don't know your anatomy, I don't know what to tell you. But once they get in here, you know, they could release these chemicals, for example, right? So what is happening is that like the active transport, vesicular transport moves these substances. Sometimes substances exit, and this is called exocytosis. All right. Uh, Sometimes material are coming in and you bring in the material. So say you have a intestinal cell, for example, and the material are coming in, you know, you moving it around, and then maybe you dumping it somewhere else to modify it, whatever the case might be. So all of these are kind of like what's known as a transcytosis or vesicular trafficking, where these substances get inside of our cells. They might get processed or stay from one region to another, and eventually they can exit. But right now, all I wanted to emphasize in exocytosis and endocytosis. All right, so let's go ahead and take a look at first endocytosis. There are different type of endocytosis, one which is called phagocytosis. Phagocytosis is also referred to as cell eating. A lot of our immune cells are like that. So here is our immune cell, for example. Here is the bacteria, for example. So, a little bit later on in time, as the time passes by, bacteria now is engulfed by the immune cell, for instance, and a little bit more in time, now the bacteria is completely inside of the cell, all right? So the cell is actually eat it up. This is not the same thing as infected. It did not get infected, all right? This is the cell came in and engulfed these particles, which in this case happen to be bacteria. This is also known as um, amoeboid motion, all right? Amoeboid is like change shape, kind of like, feet right here, they change shape, then it bringing it in, and that's what happens, okay? Phagocytosis, cell eating. Then there is a pinocytosis, also known as fluid phase endocytosis. And these are referred to as cell drinking. A lot of our intestine are kind of like that. They bring in the fluid and water inside, for example. Right, same concept. The difference is these material that are coming in are not solid. They are liquidy, okay, watery, all right? And then the last one of the type of endocytosis happen to be receptor mediated, which, as the name of it implies, I mean, that's just like, uh, you know, receptors are involved. That's pretty much that's telling you the story, what is happening. You know, receptors are involved. So let me just give you an example of that. Say here is portion of the cell. This is inside of the cell. Say this is a portion of the cell, and you might have certain receptors in here, and these are certain molecules. Say, for example, this is cholesterol. This is cholesterol receptor. Again, these are attached to the membrane. One of the function of the protein was receptor. So cholesterol bind to this receptor first. So a little bit later on in time, it will look something like that. Now the cholesterol is binded to this receptor. Now, using these receptors, you could just bring them in. All right, so they're just kind of like fold a little bit later on in time. They are folding, all right, and you slowly bring in the cholesterol inside. So you can see what is happening. It's just folded right here, all right. And again, a little bit later on in time, what happened is now is completely inside. So this cholesterol, therefore, it could get metabolized, break down, things in that, and guess what? You remove the cholesterol from your system this way. So you actually, if you have a lot of these receptors, you could actually take out a lot of this cholesterol. What if you have less of these receptors or receptors are not working properly? Well, that's the case, then you will have a higher cholesterol level in your system. So this is a great example of a receptor mediated because receptors are necessary and substances are coming in, but you need to have these receptors. Finally, there is a exocytosis. A great example of the exocytosis is what happens in the neuron. In the neuron, you're releasing the neurotransmitters or chemicals from the cell, that is the neuron or the nerve cell. And again, we can look at it in details a little bit later on, but basically, here is what happens. This is the same structure right here. The vesicles comes into the motor end plate and is being released. This is called exocytosis. All right. Uh, as far as the active transport goes, everything is required energy, ATP. We get our energy or ATP from the mitochondria. All right. Uh, well, I'm going to go ahead and talk a little bit about maybe G protein and second messenger system. I don't know if I should. Uh, Second messenger system really comes alive during our nerves. So let's cover it then. Okay, I will cover the second messenger system and how the G-link protein is involved in upcoming lectures. All right. So at this point in time, I do not want you to worry right now. Okay. Um, go ahead and
If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clavering coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
okay, you guys. So you saw that you know.
In this video, we're going to talk about something called bulk transport.
Hmm?
This process described.
Okay. So you just viewed that we could look up anything we want, and it's easy, and you guys can look things up again. So make sure to, if anything does not make sense, look at some of the animation and revisit it again. I think you'll do just fine. Okay. Next, I wanted to go over the organelle of the cell really quickly, and that will finish up our discussion. But I wanted to also give you guys some time to help each other out, to connect, to chat, to go over the materials if you wanted to do your group things and so on. So I would like to